Well, hi, I'm Clint Potter. I'm one of the founders of NanoImaging Services. Welcome to our third talk this series, this summer that launches our new facilities in Boston and in San Diego. This is a significant expansion for us. NIS has been in business for over 13 years now. Our focus has always been in providing state-of-the-art TEM services to biotech and pharma. For the first 10 years, we were primarily focused on characterization services. But in 2017, we've added a high-resolution structure capability. In 2020, we continued our major expansion by expanding our facility in San Diego with the addition of a Krios farm annex to house more microscopes. We have also started an East Coast operation with a lab and microscope in the Boston area. Between the Boston and the San Diego annex, we've added over 10,000 square feet of space to support the expansion with additional microscopes. We have the capacity for growth, and we also have a partnership with Thermo Fisher that will allow us to expand the instrumentation and keep up with the state-of-the-art in technology. Our laboratory in Boston is close to many of our clients in the biotech area, in the Boston area. We can interact directly with our clients there. And the close proximity eliminates the need to ship samples across the country, enabling us to analyze freshly prepared proteins. With our partnership program, our clients can have access to instrumentation and training from our highly experienced team. NIS focuses on TEM services over a wide range of applications, from antibody development to characterization of vaccines to high-resolution structure determination. You'll hear some more of this during Bridget's talk today. NIS is a great team. Not only that our leadership team has decades of experience, but our staff of scientists, microscopists, and analysts all strive to do the highest quality work and make your project a success. This is a final talk in the summer series for our launch, and CryoEM has advanced quickly over the past 5 years. Our co-founder and CTO, Bridget Carragher, will present a perspective on the growth of the field and a glimpse into what's in store in the future. I'll turn it over to Bridget now.
So thanks, Clint, for the introduction. And I'm going to talk a little bit about future developments in CryoEM, but I'm going to kind of give an introduction to the methods as I go along so that everybody's on the same page. And a lot of this I'm going to do with my academic hat on. As many of you will know, Clint and I run both an academic lab and we're involved in the company. We were founders of the company and still play roles in the company. But a lot of what I'm talking about, some of it's fairly cutting edge, or some of it's historical, and much of that was done in the academic lab. So you'll see the acknowledgement here to the New York Structural Biology Center and the Simons Electron Microscopy Center, which is where our academic lab is now located. So one of the first questions anybody might ask is, why do we care about CryoEM? And I like to show this slide, which shows the enormous and breathtaking growth of CryoEM over the last few years. And that's sort of illustrated. It's not the only measure, but one of the measures is the number of structures in the protein data bank, the number of protein structures that have been docked into that, submitted to the protein data bank. And you see this incredible growth here. This is a logarithmic scale. And CryoEM has just shot up, and we all know why, but I'll talk a little bit about it just to give some historical background. And it's on a trajectory to catch up with X-ray crystallography over the next 5 years, and we certainly hope it will. So that's one of the reasons we may care about CryoEM. And another reason, of course, is very well illustrated by the current crisis we're all going through with COVID-19. These are 2 papers of that dealt with the COVID-19, SARS-CoV-2 spike protein, the protein on the outside of that virus. And those structures came out 3 weeks after the gene was delivered to those groups. So that's incredibly breathtakingly fast. And of course, now there's hundreds and hundreds of more structures. But CryoEM has had an enormous role to play in both vaccine development and therapy development for COVID-19. So that's yet another reason we would care. And then finally, you've heard both from Giovanna and very much from Anette in the last webinar, all of the other things that CryoEM can do. I'm going to mostly focus today on the high resolution structure determination for both proteins and chemical compounds, but I'll come back to this at the end. Okay, so first of all, let's just say, what is CryoEM? And it's many things these days. It's of course single particle cryo-EM, the beautiful stuff that leads to these very high resolution structures. We see papers in Nature, Science and Cell every week, and lots of other journals on these. But there's another aspect of cryo-EM, which is cryo-electron tomography. Sometimes it's called that. Sometimes it's called in situ cryo-EM, sometimes it's called cellular cryo-ET. It has a number of names, and this is when you want to look inside the cell. And I'm going to talk quite a lot about that. And then there's the new kid on the block, microcrystal electron diffraction, or micro-ED, which has been incredibly successful, especially at looking at organic compounds. I'm going to quickly go through, so just in case there's people who don't know, understand exactly what these are, I'm going to quickly go through them. So just a minute or so of introduction for each. So single particle cryo-EM, that's the method we use when we have purified protein molecules or macromolecules in solution. And what we do is put a tiny little drop of that on an EM grid, usually 3 microliters. We thin it to a thin film, and we plunge that grid very rapidly into an excellent cryogen. We then have a thin film of our protein molecules trapped in this glass-like layer of water. So it's vitrified water. We take that and put that now into a transmission electron microscope. It's a transmission electron microscope. That means the electrons have to go all the way through the sample. So it's essential the sample is thin. And so what it looks like when we put those electrons through the sample is we get an image something like this. The image looks like this. And what we see are different sort of shadowy views of our proteins. Here's a stripy one, here's a roundish one. And that just is because they happen to be tumbling around in these random orientations in solution when they got snapped into the solid vitrified state. And so the electrons go all the way through these 3-dimensional structures and form projection views of the 3-dimensional structure. We also notice these images are incredibly noisy. That's because we can only open the shutter, if you like, or to let electrons onto the sample for a very brief time. If we open it for any longer, we'll just boil away the protein and do terrible damage to it. So we get around that very low signal to noise ratio by averaging. So what we do, we identify all these particles that are lying in a similar orientation, that have the same pose relative to the electron beam. We chop them out of those electron micrographs. We reorient them until they're all in the same orientation. We add them together. And now we get a better signal to noise because the signal always adds and the noise is sometimes negative, sometimes positive. So it goes away over time. If we can do that for one view, we can do it for all the other views. And we instinctively know, and also we know from CAT scanning and other medical imaging, that if we have lots of projection views of a 3-dimensional object, we can combine them to get the 3D structure essentially using tomographic methods, like CAT scanning. So that's single particle cryo-EM, and that's been around for a while. Everybody knows it quite well. What about cryo-electron tomography, or as I prefer to call it, in situ cryo-EM or cellular cryo-EM? So this comes about when the molecules you care about are actually inside a cell. Say they're inside a bacteria. You still want to know what the molecular structure is. Well, if it's a small enough cell, like a bacteria, we can use the standard freezing methods that I just described. You can put a small drop on a grid, you reduce to a thin film, you plunge freeze, and this is what that bacteria looks like when it's vitrified in your solution. You now take that grid, put it back in the transmission electron microscope, but now instead of relying on different poses of your molecules, what you do is you take that grid and you tilt it, take an image, tilt it, take another image, and so forth. And what you get is a tilt series, a sort of a loop of the thing you're interested in tilting back and forth. Well, those are projection images. We know from tomographic theory, again, that we can take a series of projection images and get a 3-dimensional volume. And here's the 3-dimensional volume of that bacteria. You can see the outer membranes here, and you can see these little dark spots. Those are ribosomes. That's great. What happens though with normal cells? That bacteria is very small. We can plunge freeze it and we can get a nice vitrified sample. But a normal cell, like this eukaryotic cell here, 50 microns, that is not going to vitrify if you plunge freeze it. And what we have to do in that case is use a high pressure freezer. And that's a fairly simple device which does just what it sounds like. You use high pressure to slow down the freezing so you can take quite a large object and freeze it without causing ice crystals. What you don't want when you're doing cryo-EM is ice. We always talk about putting it into ice. That's the last thing we put it into. It's vitrified water, in fact, but for shorthand, we often call it ice. So we put it in the high pressure freezer, and then we get a sort of slab of cells or maybe tissue even It's a big bulk object. Well, one of the questions we're going to ask is, how are we going to find what we're looking for in this bulk object? And for that, we use a device called a cryo light microscope, simply a light microscope that allows you to look at frozen hydrated specimens, usually prepared on cryo-EM grids. So when you put your sample in there, and you can use your fluorescent signal and all the rest of it, what we have in New York is a Zeiss AeriScan, then you can see the fluorescent signal and you can tell which cells you're going after, where in this big slab of cells you want to go and have a look. But the problem is, this is now a giant big bulk cell. Remember I said samples have to be very thin if you want to look at them in a transmission electron microscope. So we need yet another device. What we do is put that big block of cells into a device called a focused ion beam scanning electron microscope. And what happens inside that instrument is that, thanks to Thermo Fisher for this lovely animation, you use the focused ion beam to simply chop away most of the sample, all of the cell except a little thin window that you leave behind. And this is often called cutting windows into the cell. And so now that thin window, this is what it looks like, by the way. This is an example of some yeast cells that were embedded in a big slab, and then everything was chiseled away except this thin window of yeast cells. Now that's thin enough, we can stick it back in the transmission electron microscope, and we tilt and image and tilt and image and gather this tilt series of projection images, which once more can be turned into a three-dimensional volume. And this sort of shows a little bit how difficult this method is, because this is typically what those images look like, and it really is a sea of noise. So you may be able to make out a few things here. That's a nuclear pore complex, the portal between the cytoplasm and the nucleus. Here's some microtubules end on. Here's a ribosome in the cytoplasm. Here's a ribosome in the mitochondria. So there are objects there, but you can understand it's quite difficult to find them in this noisy environment. Nevertheless, that can be done. If you can identify them, you can go and grab them out. You're now grabbing out little 3D volumes instead of those 2D projections, but you can grab out those 3D volumes, realign them to each other, average them together, and again end up with fairly nice molecular structure. And this is a beautiful example from Wolfgang Baumeister's group. They've been doing this longest of everybody and have some really exquisite examples. So that's cryo-electron tomography, or in situ cryo-EM. And then the newest kid on the block is microcrystal electron diffraction. And in this, what we do, I'm just going to talk about organic crystals for now. If we have an organic crystal, grid preparation is pretty easy. Crush that crystal, dip the grid in it, and you see crystals sprinkled all over your EM grid. And here's an example of one centered. And then what we do is put that in the electron beam, and now we capture diffraction images rather than images. We take a diffraction tilt series of that crystal, just like the X-ray crystallographers do. We process the data, we do phasing and refinement, and we can get to atomic resolution. And I'll talk a tiny bit more about that later. And this work is very much due to NanoImaging Services side. This is not on the academic lab. So these are the pipelines we have now. So we have a single particle cryo-EM pipeline where we have to do grid preparation, we have to collect data, we have to process that data, and then of course we have to interpret it. And this works very well. We do this all the time at NanoImaging Services and of course everywhere else. I would say the only real bottleneck left is in the grid preparation side, and I'm going to talk a bit about that in a minute. On the cellular cryo-EM pipeline, the same thing. You have to do grid preparation, you have to collect data, you have to process that data, and then you have to interpret that data. You have to find things in the sea of noise and do something. And here, definitely this is a bottleneck, and definitely this is a big bottleneck. And for that reason, this is not really something we are offering. Although we do tomography at the company, we don't offer this high resolution molecular microscopy tomography because it's still a bit of a work in progress, although we hope that will change soon. And then finally, micro-ED pipeline, that works beautifully. We solve structures all the time. We've solved dozens and dozens of these structures at NanoImaging. This is for organic crystals. For protein crystals, it's a little bit more difficult. And although we can do it, I would say that's still a tiny bit of a bottleneck, and we need to improve sample prep in that area. So those are the pipelines we have. So how long has cryo-EM been around? Long time. This is the kind of origins of it. This is the 1985 3D electron microscopy Gordon Research Conference, which was the beginning for me and for many others. And there were three very important people at that meeting. You might notice Jacques Dubochet, Rockland Frank, and Richard Henderson. And they, as everybody knows, won the Nobel Prize in Chemistry in 2017 for the development of cryo-EM. So that's quite a long time ago. So why did it take so long for it to be recognized the way it is? And this is really what was happening pre about 2014. We solved lots and lots of structures with cryo-EM. They were blobs, low resolution, and the X-ray crystallographers famously called us the blobologists because we couldn't get to high resolution and we were looking at blobs. Some of them were very interesting blobs. We did lots of nice biology, but it certainly was not high resolution. And then, of course, along came the so-called resolution revolution, a change in our field so profound that I think it's very fair to call it a revolution. And this came along around about the years 2012, 2014. Our lab, our academic lab, was very much involved with this because we got one of the very first prototypes of these new instruments that came along, just as Nico Grigoriev came to spend a year on sabbatical. So we had this perfect storm of opportunity and we did a lot of work in this area. So what was this all about? What it was about was the final production of a camera that was useful to us as electron microscopists. Prior to this resolution revolution, we, like everybody else, used charge coupled devices as a camera, just the same thing that's in your iPhone or any other camera. But the problem with a CCD camera is that it wants photons. Your iPhone wants light, but we make electrons. So in order to turn those electrons into photons, we had to use phosphors and fiber optics and all kinds of things. And that turned every single electron into a little fuzz ball of photons. Then along came these direct detection devices, and they now detect electrons directly. So the immediate result of that, this was the very early work we did in about 2012 or so. If you looked at what an image looked like on a CCD, what we used to have to use compared to on a DDD, well, they look very similar, but if you look at this in frequency space, you see that the signal goes out much further on these new direct detectors than it did on the CCDs. And that's a direct result of not blurring out every electron into a little fuzzball, so get higher resolution information from these devices. But it gets even better. For years and years, we had kind of known in cryo-electron microscopy that our images were blurred, and they were blurred because when we hit this thin film of vitrified sample with a blast of electrons, the whole thing would move. The whole grid might be moving and drifting under the influence of thermal instabilities. And then these little particles themselves might be moving independent of that just because of the shock of this electrons arriving on the thin film. So these new generation of cameras don't take single frames, they take movies. Just as your iPhone does now, you can open the shutter and bang, 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 you get a whole series of movies. That's what this new generation of cameras get. So what we do is we get individual frames. This is at very, very low dose. You can hardly see anything. And then we get another frame and another frame and another frame, another frame, and so on. And we add all those together. And now we end up with the same sort of a noisy image, but at least you can see your particles, but it's made up of very many frames. So if you've got all these frames, you can page through them while you're taking them, the image, to see what is happening to your sample as it's exposed to this electron beam. And what this shows is one of the very early experiments we did. This was a 60 frame series. So we took 60 frames and then we're adding them all up together. But I'm showing you them in 10 frame samples. So the first 10, the second 10, the third 10, etc. Because otherwise you wouldn't see anything. The signal choice would be so low you wouldn't see anything. And so I'm taking those first 10, the second 10, and I'm just flipping all the way through them, 1 to 60, 1 to 60 in sets of 10. And what you can see here, there's quite a lot of evidence of motion, okay? And we can see that a little better. We'll put some fiducials down. I'm marking the edge of the substrate that supports the sample. And then these are the individual particles. These are huge particles, but we can see they're bouncing around quite a lot. We can measure exactly how much they're bouncing around because we've got these individual frames. So I can take the first frame, correlate it to the second frame, and figure out how much they moved, and so on and so on. And then I can track what is happening to my particles as I go from 1 to 60, and then I'm going to 1 to 60. I'm just flipping back through this. And what you see is they start to move, and they make huge movements. These are exaggerated vectors, obviously, but here's an accurate scale bar. And some of these particles are moving more than 20 angstroms while we take the images. So little wonder we were blobologists. We were taking grossly blurred images of our samples, and we were only getting anywhere because we took millions and millions of images and found a few that weren't moving as much, and those were basically contributing all the signal. The rest was just blur. And that's really, and so once we can do this, once we know how much they're moving, we can just correct for that movement. So we can take the first frame and align it to the second frame, align it to the third frame, and so on. And then instead of these horrible blurry images we were getting, we'll get these nice crispy images with the blur taken out. And that's what directly led to the so-called resolution revolution. This is one of the very first samples we did at sub-3 angstrom resolution in about 2014 or so. And at sub-3, you know, as you expect, you can see side chains and other details that you need to see. That's a long time ago. What we do now on a very routine basis is get sub-2 angstroms for a well-behaved specimen. That's our classic test specimen is apiferritin. It's the lysozyme of electron microscopy. And you can stick that through a microscope and in half a day get a sub-2 angstrom resolution map, and we use that to test our microscopes. So the so-called resolution revolution really gets, the cameras get an incredible amount of credit. They were the ones that came along. But we mustn't forget that for the last 30 years, a lot of other great things happened. One of which was microscopes became so much better. This current generation of microscopes is amazing. They're robust, they're reliable, they take images day after day, week after week. They don't go wrong very often, and that's a miracle, because if you ever look inside them, you'd be astonished at how many parts there are in there. And combining vacuum and cryogens and electronics, somehow they are incredibly robust and reliable. So they came a long way. And then computers, of course, we got all that for free. So the computers we were using when we did that first sub-3 angstrom map with CPUs, we spent months and months of CPU hours processing that data. Now you could do it in half an hour or so on a GPU. And we got that all for free from the fact that there are billions of kids playing games on these GPUs. So they became incredibly much cheaper. And indeed, the resolution revolution really was a function of getting better, faster, and cheaper. So the microscopes got much better, the cameras got much faster, and the computers got much cheaper. We wish all of them would be better, faster, cheaper, but you can only have one at a time, it seems. The other thing that happened was the software got incredibly good. So the software can take these very noisy images and sort out the particles from them where you can barely see the particles by eye. And one of the things it does, it can take very huge data sets and sort out the very small percentage of particles in that data set that contribute to a high resolution map. And these softwares like Relion and Krios do this routinely, very easily, and in a very automated way. They can also take a data set consisting of many different structures, lots of heterogeneous and conformational heterogeneity, and sort them into high resolution structures of many different maps. So that software I am in awe of. It has gotten incredibly good over the last 5 years and just continues to improve all the time. So the state of the art in single particle cryo-EM right now, these papers just came out this last month, they're on bioRxiv still, is 1.2 angstrom resolution. And that really is atomic resolution. Before we would say everything else is near atomic perhaps. And so what does a 1.2 angstrom resolution cryo-EM map look like? This is what it looks like, it's apiferritin. But if we zoom in, I found this sort of remarkable. That is actually a map, that is not a model. And every atom is there. It really does look like a model. And this is from that paper. You can see every single atom, you can see the hydrogen atoms. It's really a breathtaking achievement. The state of the art in cellular cryo-EM, I won't spend too much time on that because we can't really offer that routinely, but I am also breathtaken by this. This is a ribosome in situ inside a bacterial cell at 3.7 angstrom resolution with a drug, this green thing, clearly bound. So this is a drugged ribosome inside a bacteria that is visualized at sub-4 angstrom resolution. Again, a very remarkable achievement, and I think that's still also on bioRxiv. And then I won't call this state of the art, but it's certainly very achievable and almost routinely in our company lab now, and that's to do the micro EGM. This is one that Jessica and Giovanna did recently. It's donipicide. It's fairly large for micro EG, 660 Daltons, solved to 1.15 angstroms, and then solved de novo to get the atomic structure. Okay, so that's all the good news. What about the bad news? So we have this very nice pipeline, but I told you there was a bottleneck. The grid preparation, why, well, you might say, well, why isn't everybody at 1.2 angstrom if apoferritin can get there? And one of the reasons is the sample doesn't always behave itself, but one of the reasons is grid preparation. And so there are many issues with grid preparation. Sometimes your particle will behave beautifully biochemically, look great in stain, you put it in ice and it's an aggregated mess. Sometimes when you put it in ice, all your particles will stubbornly adopt a single orientation. And if you can only see one orientation, it's hard to get a 3D map. Sometimes the particles will stick to the support film and they won't go into the holes where you want to look at them. And so you're stuck. Many times particles are partly damaged. I said earlier, we only sometimes 5% of the particles go into the map. Why is that? And then sometimes you don't see any particles at all. They completely denature when you put them into ice. And there was lots of debate about this over the last few years, but I think it's quite clear, we all understand now that what plays an enormous role is the fact that our particles are at an air-water interface, and proteins do not enjoy being at an air-water interface. So, what we visualized, our beautiful particles all perfectly lined up, in the middle of the water, safe and sound, what really happens is the particles end up stuck to one or both of the air-water interfaces. And that's because we have to thin our bulk solution down to a very thin film in order to vitrify it. And while it's in that thin film, the proteins are moving under Brownian motion and banging into the air-water interface. And if they have any propensity to stick to that air-water interface, that is what they'll do. And that's where they end up. We're not making this up. We know this for sure because Alex Noble, a postdoc in our lab, went and did tomography on every single particle data set he could get his hands on. He ended up doing 50. And in 99% of the cases, what he found, no matter what the sample was, the proteins are stuck to both or one of these air-water interfaces. So we know for sure that's where proteins end up. So that's one reason we want to improve sample preparation. And the other reason is more sort of fundamental. I told you the way we make grids is we have proteins bumping around in solution and we snap them into a thin film. But the way we do that is we dump 3 microliters of sample onto a grid, and then we smush 2 giant pieces of blotting paper onto the grid, and we remove 99.99% of it, and we throw it into the garbage can, and then the tiny little couple of nanoliters that are left end up on the grid. And the grid can be sometimes too thick, sometimes too thin, sometimes damaged, and that's because we're using a very brute force macroscopic method to try and make a nanoscopic sample. We really want this thing to be about 100 nanometers thick. So it's not a great method for doing this. So quite a few years ago, Clint and I decided that this was really the step we needed to automate next. We'd automated data collection, automated processing. What about improving grid preparation? So we knew we needed 2 technologies. We wanted to not throw away so much sample. So we decided to use piezoelectric dispensing devices. And here's an example of what we're doing. This is a piezo dispenser dispensing a 50 picoliter droplet of protein out of its tip. Remember, on this method, we use 3 microliters. So we're going from micro to nano to pico. So this is 1,000 times 1,000 that we're dropping in volume in order to do this. So we don't want to waste all our precious protein. And then we'd like to improve this macroscopic method, trying to make nanoscopic. And we did that by inventing and developing so-called self-blotting nanowire grids. And I'll quickly explain what those are. So the basic idea we have is a piezoelectric device. This is it here. That's the tip right there. And here's a video showing with strobe lights the protein sample, the 50 picoliter spot of protein spitting out. And all we do is we take the grid and we race it past this tip while the tip sprays out droplets onto the grid. And here's like a continuous loop of that happening. There's a line of droplets sprayed across the grid as it races past on its way to the liquid ethane. And the way we get it to spread out into a nice thin film, not be a big blob, is we have these so-called self-blotting nanowire grids. And all those is a copper grid that you put into a very simple chemical for a couple of minutes. You take it out, wash it, and it's covered in this beautiful bowling green lawn of nanowires. And those nanowires act now as very nicely controlled blotting paper. And so this is the setup we have for our device. Here's the sample being spat out. Here's a grid with a substrate to hold your liquid when it hits there. And these are all surrounding it, the nanowires. And here's a video showing what happens when that 50 picoliter drop hits our nanowire grid. And I've also taken a still frame to kind of illustrate what is going on during that process. So the drop hits the substrate, starts to spread out under its own momentum, spreads out a bit. But then the instant the water comes into contact with the nanowires, capillary action just sweeps it very, very rapidly away, leaving a very thin film behind. And that's what Spinodon does. Here's an example, a couple of test examples. I can't remember what this is, elderles maybe, and here's apiferritin again. And so we have a swish of droplets across the grid, making a very thin film with beautifully thin ice, nicely distributed, and we can get high resolution structures from this. And we did these as proof of concept, because you always should do a proof of concept, and because every single person we told about this project at the beginning assured us it would never work because the proteins would be destroyed by the piezo device. So sometimes you should just try things anyway, and you'd be surprised that they succeed. The other nice thing about this project, about the Spinodon project, is that because the grid is racing past the sample on its way to ethane, the sample does not spend too much time in that thin down film of water. And as a result, if you use this device, more of the sample is in solution and not in contact with the air-water interface. So it has a sort of good effect on many of those problems we talked about, like preferred orientation, like protein denaturation. And these are some of the many samples that have been prepared using this device, using Spinodon, and been published. I mean, here's one from Giovanna when she was working in our academic lab with insulin bound to insulin receptor, and that problem was that it was a highly preferred orientation. We used Spinodon to disturb that. Here's a beautiful example from Carolyn Lugar's lab where this protein just completely denatures if you put it into a thin film using standard Vitrobot or other cryo preparation devices. So those are some examples of that. The Spinodon project has now been commercialized. This is what we did in 2011. We visualized that this was our beautiful device. This is what it really looked like, a bit of a, the Rube Goldberg device, but it worked nevertheless and still does. And now it's been commercialized by the company TTB LabTech, now called SPT LabTech. That's their beautiful visualization, but it is a really beautiful device, and it's highly automated and does a really good job. So I'm really hoping these devices move into more and more labs so that companies and people who want to do this can simply make grids by themselves very close to the production of the protein and then ship them to people like us for imaging. So just to wrap up then, the single particle pipeline works really well, solves many, many structures. We solve a ton of structures at NIS. Of course, there's many, many being solved in academia. And I said there was a bottleneck with grid prep, but I hope with the rollout of devices like Chameleon, and there's many other people working on other devices to simplify and improve this process, that soon that bottleneck will be greatly relieved and reduced. For the cellular cryo-EM pipeline, these two bottlenecks certainly exist still, and it makes it hard to do in a very routine way. But I think over the next few years, automation will solve this one. People like Thermo Fisher are already automating the instruments to a large extent. People are writing software to cut these samples and do all kinds of other fancy things and developing all kinds of equipment to make that better. So I hope over the next 5 years, we'll see a lot of that. And then I'm very optimistic that machine learning will make a huge impact on the interpretation of this data, be able to go in and hunt down those individual macro and molecular machines, pull them out and do the averaging and 3D reconstruction and make that much more of a routine process. And then the micro-ED pipeline, I mentioned that proteins are a little bit more difficult to do than the organic crystals, but I think, again, you know, devices like Chameleon, like other sample prep methods will get us over that hump so that we can do that in a more routine fashion as well. Okay, so at NanoImaging Services, we certainly do micro-ED and we certainly do a high resolution structure. And then you heard last week from Anette, we do all kinds of other things too. Vaccine and virus characterization, delivery vehicles, we do antibody characterization. So there's many, many different ways that you can use cryo-EM in all its different forms in order to answer questions and get results. And it's quite clear, I think, to me and hope to everybody else, that this entire field has an incredibly bright future, and we'll just go from strength to strength and be adopted in a wider and wider fashion until everybody is using it as a routine method. Thanks, I'm done. And I think, shall I hand over to Mel to handle questions and things?
Yes, please.
Okay, I'll stop sharing. I hope. And I stopped sharing. Yes, okay.
Oh, there we go. The full complement is here. For any of the participants, if you would like to continue to submit questions through the Q&A panel at the bottom of your screen, please go ahead. And we've had some that have come in actually in advance of this session. I think there's been some good food for thought. Let's start with a couple of those. So in terms of protein dynamics, can we comment on how that's handled in cryo-EM, particularly between domains or maybe the local dynamics of flexible residues?
So you sort of are asking the question about flexibility. And I mean, I think the very best thing about cryo-EM is that it can handle flexibility. You'll often find you get a map, some of it is at very high resolution and some of it's not at such high resolutions. So there's two things to be said about that. One is some of that not high resolution tells you a lot. It answers a lot of questions about what is your protein up to. And the second thing is we can sometimes focus in, do a method we call focus classification, focused in on the dynamic section and improve the resolution in that localized area. And then you sort of patch your protein back together. But I think not only can you kind of try to solve that problem, it also teaches you a lot. And, you know, I think for the crystallographers in particular, a lot of these targets, drug targets, are going to be looked at cryo-EM, analyzed and understood. Where are the flexible bits? Where are the solid bits? And then the crystallographers can start chopping it up until they have the bit they're interested in as a solid dynamic section. And then they can go crystallize that and look at it with 10,000 targets. So I think crystallography, cryo-EM are going to work hand in hand to solve these problems and make it better for everyone. Anybody else want to add to that?
Well, I guess from a practical perspective, how much flexibility or compositional variability becomes too much, particularly for some of our drug discovery groups that are looking to enable high resolution structures?
Does Giovanna want to answer that question? Because she's actually done this in practice, so she'd probably be a better, and probably people are sick of me talking by now. Go for it, Giovanna.
So one thing I learned is that it's not just how much flexibility is there, it has to be a discrete flexibility. And so we have to be able to separate different classes that have different conformation. And if that's the case, I think we can handle very complex systems. As long as we can classify all the different 20 conformations, we can definitely solve the problem. If what we have in solution is like of a continuum of different conformation, then it becomes much more complicated because we can actually separate them. And so that is a no-go basically for our purposes. I've seen structures with 20 different conformations, and all 20 of them have been easily solved, collecting the right number, large number of data, but it's definitely doable.
I knew Giovanna was the right one to answer that.
So we've had an inbound query just now on how much does a chameleon cost? We're going to defer that to SBT Lab Tech. My response, for those of you who are familiar with my background, is more than maybe we had planned. But we hear there are lots of bells and whistles, so we're looking forward to getting our upgrade. In terms of sample prep on the chameleon or with other low volume dispensers, You know, when we start to think about piezo-based dispensers, are there special considerations for samples like membrane proteins that may have different solubilization agents, or can it be broadly applied?
Broadly applied. We were astonished, actually. We thought we might struggle and, you know, it would spit different or have to be fussed with, but no. I mean, things with glycerol in, things with detergent in, soluble proteins. We've even, you know, dispensed bacteria and things. So I think, you know, people think of 50 picoliters as a small thing, but it's actually quite a large thing. It's 30 microns across, and your protein's at 100 nanometers, you know, best, that's a huge one. So it's still quite a large volume compared to your protein, and it doesn't seem to bother. You change the parameters of the spitting a little bit to cope, but otherwise it seems to do fine. We've done, I don't know, 50, 100 different proteins.
And I think continuing from that, maybe in terms of thinking of now stabilizing membrane proteins, any comments from anyone on the use of liposomes for stabilizing membrane proteins, particularly with Fred Sigworth's recent work on structure determination of membrane proteins from liposomes?
Do you want me to do that, or does somebody want to jump in? Otherwise, I don't mind doing it. We just did a journal club yesterday, actually, on this latest paper. I think it was in PNAS, where they got to 3.7 angstroms with a protein embedded in a lipid membrane. It's not easy, that's for sure, because the liposome is big and the protein is small. So you're looking at quite a thick layer of ice because that's got to sort of sustain the liposome, but your protein is quite small. Normally, you know, we want the ice as thin as possible. But having said that, you know, there's this new beautiful work at 3.7 angstroms. Aha, thanks, Clint. I bet that was Clint. So it's very beautiful. It was quite a large, well-behaved protein. So you could pull it out of that liposome and average it, etc., fairly well. I think if something was really embedded in the lipid, it would be very, very difficult. But, you know, having said that, 5 years ago, I would have said almost everything we do now is impossible. So we should be optimists because we've seen an enormously optimistic change in our field.
So from a bit of a practical perspective in working with us, we've had a question as to whether someone could send grids from outside the US to NanoImaging, and maybe we can expand that out in terms of, for those people in the audience who have grids available, how do they get them to us? How do they work with us? What does that look like?
I think Anette should answer that, yeah, or Jeff.
Yeah, this is certainly possible. I mean, lots of clients send us grids from within the country, and there's no reason why you couldn't send grids from outside the country. You know, dry shippers hold their temperatures for many days. So even if it takes a couple of days for the dry shipper to arrive, that is not an issue. That's actually quite a straightforward process for us. The only thing, you know, obviously we have a bit of a conversation with the client before they ship their grids to find out a little bit more about, you know, what is the protein, how were these grids made, what exactly is the goal, is this like a screening project, a Krios data collection project. We'd have a very simple grid submission form where we ask the client to fill it out prior to submission of the grids, but it's really quite a straightforward process and should not be a problem at all from outside the country, outside the US.
And in fact, samples we certainly get sent from India, from Japan, from Europe, from everywhere. They arrive constantly. We're used to dealing with that.
Mm-hmm. And maybe one for Jeff. I'm going to put him on the spot because he's quiet at the bottom of my screen. For clients who are actively collecting data with us, what are the options? Is it synchrotron type for those crystallographers out there, or do we just by default do everything? What's available?
Yeah, one of the great things about our company is our experience from all the way from the sample to the data collection. So we're able to support you at any particular part of the workflow for getting your sample to a grid and into imaging. So if you have grid making capabilities and you want to send us grids, we can do like a synchrotron style data collection where we take in your grids, load them up on our scopes and start the data collection. And one of the nice things we do is we have a live online viewer, so you can actually watch your images come in real time and actually provide feedback or have a conversation with us as to what the optimal parameters would be or optimal situation is for your data collection. If you don't have those capabilities or you want us to give it a try, you can send us your sample and we'll go ahead and try to make the best grids possible. We have a lot of experience in different methods and different instruments for doing this. And we can make the grids and move forward from there in that model.
Great. Back to sample prep. I think you've scared some people, Bridget. You know, if somebody doesn't have a next generation sample prep system available and they're experiencing issues with air-water interface effects, can you comment on some of the other strategies? And I'll loop in another question with that in terms of surface functionalization approaches and any benefits there in sample prep.
Yes, there's enormous amounts of work being done on this at the moment. And in fact, that little liposome paper Clint just showed, those liposomes were laid down onto graphene. So you want, the bottom line is you want to keep your sample away from the air-water interface. What do you do about that? Well, Spinodon or chameleon is one thing. You go so fast, it doesn't have so much time. Another thing is, why don't you put some detergent into your sample, and maybe the detergent will coat the air-water interface, and when your sample bumps into it, it won't be so inclined to stick. Another thing is, let's put a substrate down and pull the sample onto the substrate so it can't get to the air-water interface. And for that, people have often used graphene or graphene oxide. One of the problems with graphene or graphene oxide is, well, now your sample's not sticking to the air-water interface, it's sticking to the graphene or the graphene oxide, and that might be bad. So then maybe you can functionalize the surface of the graphene with a long loopy thing, and the protein is caught onto that, and it's not sticking to the graphene, it's just sort of floating in mid-air, but still sort of, you know, tethered a little bit. All of these things are being done, have been done, have promise. I would say so far nothing works 100% of the time, because if it did, every single paper you see in the literature would be using that method and we wouldn't use any other method. But what we do, as Jeff just said, at NanoImaging Services, we'll try all of the above. We're having problems, we'll try the detergents, we'll, you know, try various additives, we'll try changing biochemical conditions. And, you know, I'm not a huge fan of graphene yet because I think it's a little bit unpredictable, but that would be also something to try. So yeah, there's lots of things you can do. Not all of them work every time.
For smaller biotechs that are outsourcing their upstream expression and purification, is it still possible for them to work with nanoimaging? And what would that process be? And maybe we can comment a little bit on things that someone should ask of them in sort of providing them with insight as to what a cryo-EM grade sample would be.
You're probably the best person to answer.
I was just going to say, I think, Melanie, I think you're the best person to answer that question.
I completely agree, Mel. We're putting you on the spot.
Three of these without having to answer anything. The truth is, we're happy to work with any upstream providers, and we can do so quite collaboratively, starting as early in the process as you choose. If you'd like some recommendations for groups that we've worked with before based on the target class, we can do that. And we're happy to provide, you know, input along the way as one of your partners to help you identify a good provider, provide guidance into best practices, and let them know specifically what some of the challenges or bottlenecks might be in generating a cryo-EMable sample. And Giovanna's worked with a few providers herself, so she will probably wind up on the phone with you to help. And following from that, we've just had a question. Before we try to put samples on grids, are there quality assurance, quality control tests? Are there recommended biochemical and biophysical profiles that we look for before we suggest that a sample is ready?
Yeah, I mean, this is an excellent question, and it is something that always comes up, and the answers are a little bit sort of not very, I guess we don't have a really, really good answer, but some things that should be done for sure is you want to have an idea that your particles are monodisperse. A good thing to test that out is on a, let's say, size exclusion column. I think everybody does this. you want to have a sense of the purity of the sample that can be checked by gel electrophoresis. Everybody does this. These are sort of routine, very simple things. And then there, you know, then you ask now, okay, what else could be done? Because oftentimes, you know, somebody comes along and says, I have an excellently sharp peak on my SEC profile, my sample is good, and we find it's not so good. So what else could be done? You know, we've talked a lot about, you know, things like testing whether freezing and thawing affects the sample. You know, how does it behave if it's frozen and thawed again? Is it still behaving the same way in terms of dispersity, or does it start to aggregate? There's also this new, you know, this instrument Refine. We've talked a lot about it and how that can provide some really interesting information about a sample's behavior and composition. But I think, and others can chime in here, I'm probably forgetting other sort of approaches that people can use to test their sample. It is still a lot of, yeah, it's still kind of unpredictable, really. And every case is a little bit different. But these three things that I mentioned, they should at least, you know, they should be part of the quality control before a sample is submitted. I don't know what else.
There are some other services that we're looking at to try and introduce that would improve the QC steps. So we recently launched a negative stain screen, which is basically just a very quick look-see at your sample to see if it's worthy of going through to the next steps, which is you know, a low-cost service. And obviously, if people can do, you know, similar kind of like QCs in their own labs before they send things to us that we can match up, those are really the best things that we can do, things that we can replicate. And that really will help improve the QC on both sides.
Plus,
Maybe Giovanna wants to comment too, because you were in the trenches doing this.
So I always think that the biochemist or the person that makes the protein is the person that best know the protein or the sample. And so you know what it's required to make the protein happy. And you know what are the tests and the things that you want to run on the protein itself to make sure that it's a happy protein. So one thing I would definitely recommend is to, if you have a ligand that you know stabilizes the protein, try to use that, because the least amount of flexibility it's present, the least amount of problems we can have down the road. If you're looking for something that is in a conformationally active, in an active conformation, make sure that what you give us is active in that active conformation, because very often we end up solving a structure or something that doesn't mean anything to the client because it's not what they were looking for. It was just what looked the best on gel filtration, but not necessarily what they were looking for. So use your biochemical knowledge on the sample to make it good, and then run these few tests. They're not, as Anette said, they will never give you the absolute answer. We will have to try and put it in ice before we can actually say something. But they do increase the chances that you go in ice.
And turning to some practical logistics, we've had a question of how many microscopes and what kinds do we have and where are they located? That's just come in on the Q&A. So for those who probably missed the overview on webinar 1, if somebody wants to give a little bit of a rundown of where we are and where we might be in the next couple of years.
I can do that. So we have, you know, offices in San Diego. Actually, we have 2 locations in San Diego. We just put in a new annex so we can really expand the number of microscopes we have. So in San Diego, we have 2 big high-end Krios microscopes, as well as a Glacios microscope and a smaller screening microscope called a Tech 912. And then we recently expanded to the Boston area, where again, we have a Glacios microscope right now, but again, we can expand there as well. You know, it depends. I mean, if you really need microscope time, we can probably accommodate you even fairly quickly. So, you know, ideas, contact us and we'll see if we can help you. We do have the ability, you know, to really expand our capability in microscopes fairly quickly with a partnership that we do have with Thermo Fisher. So right now we're not really constrained in terms of, you know, microscope availability or space for them. We can expand, you know, to quite a few microscopes.
One thing for sure, NIS is very quick. I know a lot of clients work with academic labs. Those academic labs, we've heard, can be very slow, and that's because academic labs have other fish to fry. You know, they're doing research and they've got grad students and postdocs to take care of, etc. What we take care of is our customers, and turnaround time can be very quick. Almost every project is turned around in 3 weeks, and we can do rush jobs if you want. So turnaround time is not something that anybody ever complains about with NanoImaging services.
And the idea too, I mean, if you're collecting data on one of our Krios, for the idea you can see your data coming in the way you have things set up. And
it's shipped to you immediately. It's shipped in real time, so you can start processing it in real time.
And I think we're down to probably the last question, which is, can we expect other regions to have local NIS facilities? And what kind of microscopes can we expect for them?
I don't know. And the reason I'm just basically, we went to the East Coast. We doubled our number of staff. We added 2 new facilities this year, all during the pandemic. So we have a team that was able to do this. But if you ask me right now what we wanted to do this, I would hedge. But realistically, Boston is a trial with the idea of, if we're close to our customers, like the biotech and pharma community in Boston, does that really help you? Can you get us samples right off the column, that thing? You don't have to ship it across the country. So, I mean, the obvious next step would be the San Francisco Bay Area for us.
And Clint's such a pessimist, and I'm always the optimist. And I'd say, yes, next we're going to South San Francisco. And after that, Philadelphia. And then, so I would say if this all works out, obviously we wouldn't do that. We just need to get it all working. Send us your samples and your grids and we'll see how we go.
Great. And with that, I'm going to quickly answer these other couple of questions just here on the chat, and we'll wrap up. One question is, do we have a second on site to monitor quality? At the moment, we don't. A lot of our clients do use SecMols to profile samples before they're sent to us. As Ben mentioned, we are considering, adding other possible upstream instrumentation that would help us either monitor QC or provide additional polishing steps. So if that's something that you think is of high value for us to offer, send us a note, send in your votes. We're getting ready to fill some bench space over the next few months in these new facilities. So we certainly want to be able to continue to expand services. The other question is, do we charge by successful images or by hours and efforts? At the moment in the microscopes, we really, for the most part, charged by a period of time. And that includes FTE access and all the bells and whistles that the team has described. We do have some fee-for-service work that's really based on incremental projects as opposed to large microscope bookings. If you go to our website and the Working With Us section, you'll see all the different ways you can possibly engage. And that's really just because it's a rapidly evolving area and all of our clients really have different needs, expectations, internal infrastructure, workflows, et cetera. So we've tried to make everything as customizable to each client's needs as possible. So with that, I think we will wrap up the session and the last of this series of webinars. We may be back in the fall, depending on how much energy we have for some Zoom webinar scheduling in the next couple of weeks. And in the meantime, please feel free to reach out to us at either info@nanoimagingservices.com or through the contact us button on the website. We'd love to have a chat with you about current projects, future projects, or just to generally learn more about where you are as a company in structural biology and drug discovery and development. So thanks everybody for sticking with us fairly late in the evening on the East Coast and for those overseas, and we hope to talk to you soon. Thanks everyone.
Thanks, bye.